We all know our bodies are always like renewing and replacing cells. But like, have you ever thought about those tiny parts inside them? You know, the proteins, especially when it comes to something as important as a woman's ovaries. We're talking about cells that might need to last like decades.
Right. It makes you wonder, like, how do those proteins in a woman's eggs manage to, stay functional for her entire reproductive lifespan?
Yeah, it's a real endurance test at the cellular level. It's amazing. Please don't forget to like the video and subscribe if you want more content like this.
Absolutely. And there's a super cool study. It was published in Nature Cell Biology that really dives deep into this. They used like some really advanced techniques, and what they found out about how long proteins can actually last in the ovary is pretty mind-blowing.
So that's what we're going to be like exploring today.
Yeah.
Trying to understand what this means for, female fertility and aging.
Exactly.
Okay, cool. So let's start by zooming in on the oocytes themselves. Those are the egg cells, right? Mm-hmm. So the researchers, they did this really neat thing called a pulse chase experiment. Basically, they tagged a bunch of molecules and then saw how long those tags stayed around.
Right. It's like a molecular tracking system.
Totally. And in this case, they labeled all the proteins in developing mouse oocytes with this special form of lysine. It's an amino acid. And they called it sub-13, sub-6 sublysine. And they did this while the mouse was still in the womb.
Yeah.
Right? And then after the mice were born, they switched them to a normal diet with regular lysine. This way they could track how long those original labeled proteins were hanging out in the oocytes, as the mice got older. Pretty clever, huh?
Oh, yeah. It's a very elegant approach. And the results were, pretty remarkable.
What did they find?
Well, even after 8 weeks, which is a good chunk of a mouse's life, they found that out of over 7,000 proteins, a whopping 768 of them still had that original label.
Whoa, that's a lot. So does that mean those proteins were there from the very beginning?
Yeah, pretty much. It suggests that those proteins were present from birth and hadn't been, replaced or broken down.
That's wild. Imagine finding original parts in a machine that's been running nonstop since day one.
Right.
And these weren't just any random proteins, right?
Yeah.
These were some pretty important ones.
Oh, yeah, for sure. I mean, we're talking about proteins like Twinkle.
Twinkle, twinkle, little star.
Oh, no. Haha. Twinkle, it's a mitochondrial DNA helicase.
What?
A helicase. It's an enzyme that unwinds DNA.
Sorry.
And it's really important for replicating the DNA inside the mitochondria, the powerhouses of the cell.
Right. Mitochondria. Got it. So what else?
Well, there's also TTC19. That's a vital part of the mitochondrial respiratory chain. That's the process that makes energy for the cell. And then there's BOP1. That one's essential for assembling ribosomes. Those are the protein factors in the cell.
Okay.
And then there's HIRA, which is a histone chaperone. It helps organize DNA by wrapping it around these proteins called histones.
Oh, interesting. So it's like a DNA organizer.
Yeah, kind of.
And you also mentioned the cohesin complex with things like SMC3 and SMC1A and PDS5B.
Yeah.
Those are really important for organizing chromosomes, right?
Yeah.
Especially during cell division, making sure everything's lined up correctly.
Absolutely. They're the traffic cops of cell division.
Uh-huh. I like that. So basically what we're seeing is that these long-lived proteins are involved in some pretty fundamental processes, really crucial stuff for the oocyte to function properly.
Yeah, that's the key takeaway here. They're not just random proteins hanging around. They're the essential workers of the oocyte.
I see. And is there any reason why these specific types of proteins seem to be favored for this super longevity?
Well, the researchers found that this group of long-lived proteins in oocytes, they were really enriched in mitochondrial proteins, like we've been talking about, and also proteins that help maintain proteostasis.
Proteostasis, was that?
It's basically the cell's system for making sure its proteins are, in good shape. It involves making new proteins, folding them correctly, and getting rid of any damaged ones.
Oh, like a protein quality control system.
Exactly. So what this enrichment tells us is that oocytes seem to prioritize keeping their energy production and protein quality control systems in tip-top shape for, the long haul.
They're built for the long game.
Exactly.
Okay, so we've been talking a lot about oocytes, but what about the rest of the ovary? It's not just egg cells in there, right? There are other cells too.
Right, there are the somatic cells, the supporting cells.
Yeah, like the granulosa cells, stromal cells, the CETA cells. Did the researchers find a a similar pattern of protein longevity in those cells?
They did, and it's a really interesting part of the study. To figure this out, they did 2 more pulse chase experiments, but this time they looked at whole ovaries. One experiment was the same label until birth approach they used for the oocytes. The other one involved labeling the mice until weaning so they could capture proteins made before and after birth.
Got it.
And then they used some pretty fancy mathematical modeling to determine the turnover rates or half-lives of the proteins in the ovary.
So, just to remind everyone, a protein's half-life is basically the time it takes for half of that protein to be replaced.
Right. And while they found that the average protein half-life in the ovary was around 8 to 10 days, which is similar to other organs.
Yeah.
The really cool finding was that a much higher percentage of ovarian proteins, more than 10%, had what they called extreme longevity. That means they had half-lives of over 100 days.
Wow, over 100 days. That's like way longer than the average.
Right. And to put that into perspective, in tissues like the brain, muscle, or even cartilage, less than 1% of the proteins had this kind of super stability.
Wow, that's a huge difference. So it sounds like the ovary is like playing by its own rules when it comes to protein stability.
Yeah, it really does. It's a unique environment.
I see. And were there any differences in like protein longevity within the ovary itself? Like between different types of cells?
Yeah, they did notice some interesting variations, like the mitochondrial proteins in the whole ovary. They also tended to have longer half-lives compared to the average ovarian protein.
Okay.
Which kind of makes sense given what we saw with the oocytes, right?
Yeah.
And interestingly, the proteins in the nucleus, they generally had shorter half-lives.
Oh, that's interesting.
They also found several protein complexes, those are groups of proteins that work together, that had many long-lived components.
Oh, okay.
Like the cytoplasmic ribosome that's involved in making proteins.
Right.
And the proteasome, which breaks down old or damaged proteins.
Okay.
And the spliceosome, which processes RNA. It's like the ovary is really trying hard to keep these essential genes together for as long as possible.
Right. It makes sense.
They even, they grouped the ovarian proteins based on their longevity, and they found this cluster of like 352 super long-lived proteins, and these guys had an average half-life of, get this, 681 days.
There's 681 days. That's almost 2 years.
Yeah, in a mouse, that's practically a lifetime. And some of these proteins, they were around for almost the entire lifespan of the mouse, which is about 15 months.
Crazy. So what are some examples of these like super durable proteins they found in the whole ovary?
Okay, so again, a lot of mitochondrial proteins popped up, things like cytochrome C, TOM-M70, which helps import other proteins into the mitochondria, MRPS9, part of the mitochondrial ribosome, MDH2, which is an enzyme involved in energy production, and GLED1, another enzyme.
Okay.
And they also found a bunch of chaperones and heat shock proteins like DNHJC7, DNAJ1, and a bunch of HSPAs. These guys are really good at preventing proteins from misfolding and helping cells deal with stress.
Right. So it's not just about like keeping those proteins around for a long time. It's about making sure they're still working properly, right?
Mm-hmm. Exactly. For example, DNAJA1, it's been shown to protect cells from apoptosis. That's programmed cell death.
Oh, wow. So these long-lived proteins, they're not just relics of the past. They're actively working to keep everything running smoothly and protecting the ovary from damage.
Yep, that's exactly it.
Amazing. And what other long-lived proteins did they find?
They found SMCHD1, which is an epigenetic regulator. It basically controls gene activity without changing the DNA sequence itself.
Okay.
And it's involved in X chromosome inactivation. And they found other epigenetic and nuclear transport proteins like DNMT3A, HDAC1, RAN, and a few others, and even some myosins. Those are usually involved in muscle contraction and cell structure, but even some of them showed this incredible longevity in the ovary.
So many different types of proteins, all with their own important jobs.
Right. It's a whole intricate network.
Yeah, it's incredible. It makes you appreciate just how complex, like, cellular processes really are. So we've seen that the oocytes have this strategy of, like, holding on to proteins for a long time. But what about the other cells in the ovary, the ones that support the oocytes? Are they in on this too?
Yeah, that's a really important question. And the researchers found that some of these somatic cells, the granulosa cells, stromal cells, and the Titica cells, they also express the genes for these long-lived proteins.
Oh, interesting. So it's not just the oocytes themselves, it's their support system too.
Exactly. It's like a team effort. To really confirm this, they used a technique called nanosims. Basically, it lets them look at the isotopic composition inside individual cells so they can actually see which parts of the cell were made with the labeled building blocks from way back when.
Wow, that's high tech.
Yeah. And what they found was that in some stromal cells, granulosa cells, especially those around small follicles and the endocells, they had high levels of that SUP13 SUPEC label.
Meaning they were like really old.
Well, it means they contained components that had been around for a very long time.
Okay, I see.
And interestingly, the cells of the corpus luteum, which forms after ovulation and doesn't last very long, they didn't show the same longevity.
So it's like the cells that are involved in like nurturing the oocytes early on, they're the ones that are built to last.
Yeah, it seems like there's a coordinated effort within the ovary to maintain cellular components for a long time. And it involves different types of cells.
That makes sense. Now, if these proteins are sticking around for so long, is there a chance they might, get damaged or clump together over time?
That's a great question. And the researchers actually looked into that too. They wanted to see how protein quality control, or proteostasis, was affected in oocytes from young and old mice.
Makes sense. Like, if proteins are hanging around for ages, you'd think that there might be some problems with them misfolding or forming aggregates, right?
Exactly. But surprisingly, what they found was that the levels of protein aggregates, those are called agresomes, they didn't increase with age in oocytes.
So they're staying in good shape.
Yeah, which is different from what you see in other aging tissues like the brain. And they also checked the activity of the proteasome. Remember, that's the cellular machinery that breaks down damaged proteins. And they found that its activity didn't decline in older oocytes. In fact, it might have even been a little higher.
That's interesting. So it's like oocytes have this really robust system for maintaining protein quality, even as they get older.
They do. However, that doesn't mean that the aging ovary is completely immune to changes. The researchers did this big proteome profiling study using a technique called DIAMS, which lets them identify and measure thousands of proteins at once.
Okay.
And what they found was that even though those long-lived proteins are maintained for a long time, the levels of many of them do eventually go down as the ovary ages and fertility starts to decline.
So even though the proteins themselves are super stable, the ovary eventually has trouble keeping up with, like, making enough of them.
Yeah, that's the idea.
So which types of proteins were most affected by this decline?
Well, they found these clusters of proteins that were really abundant in young, fertile females, but their levels dropped with age. One cluster was full of proteins involved in important things, like stem cell division, maintaining telomeres, those are the protective caps on the ends of chromosomes, assembling heterochromatin, which is a tightly packed form of DNA, and repairing DNA double-strand breaks. All really essential stuff for keeping the germline healthy.
Yeah, for sure.
And another cluster that declined with age included proteins related to autophagy, that's the cell's recycling system, chaperones, which help proteins fold correctly, zona pellucida proteins, those form the outer layer of the egg, and cohesion proteins, which are involved in chromosome organization.
So it's like the very systems that are so well maintained early on, they start to show some wear and tear as the ovary ages.
Yeah, and this seems to happen right around the time that female fertility starts to decline naturally.
Makes sense.
And importantly, they found that even many of those super long-lived proteins we talked about earlier, they also decreased in the aging ovary.
Oh, so even the most durable ones aren't immune.
No, not completely. And this included proteins involved in proteostasis, like chaperones, proteasomal proteins, and ribosomal proteins, as well as proteins involved in organizing heterochromatin.
So it seems like this strategy of extreme protein longevity, while it's good for maintaining things for a long time, it might also have a downside. Like, it helps preserve the germline for a while, but eventually the ovary can't keep up with replenishing those essential proteins, and that contributes to aging and fertility decline.
That's a great summary. It really highlights the trade-off that's going on here. The mammalian ovary has evolved this amazing ability to hold on to proteins for a long time, probably to protect the female germline. But this study suggests that even these super stable proteins eventually become depleted, and that's linked to reproductive aging.
It's fascinating. And it makes you wonder about what's happening in other parts of the body. Like, we've seen that protein longevity can vary a lot between different tissues. So what other strategies might be at play in other organs? And could learning more about these different mechanisms help us understand how to, like, extend human health span? It's pretty mind-blowing stuff to think about. Please don't forget to like the video and subscribe if you want more content like this.
I agree. It's a really exciting area of research.
thanks for joining us today. It was great to dive deep into this with you.
You too.
And everyone listening, thanks for tuning in, and we'll catch you next time.